Okay, so the second half of our DNA and biotechnology unit is going to deal with gene regulation. Much of what we know about how genes are turned on and off comes from our study of bacteria. Now, why would that be the case? Well, number one, bacteria are much smaller, much simpler, so it's simply easier to study how genes are turned on and off in bacteria. So I want you to think about, in our body, for instance, we have 46 chromosomes with thousands of genes on each and every one of those 46 chromosomes, and that same exact DNA is in every single one of those trillions of cells in our body. So the same recipe for making the same proteins is in every single cell of your body. But does every single cell produce all of those thousands of proteins? Well, the answer is no. For instance, if we think about the production of breast milk, you gentlemen will never ever produce breast milk in any of those cells in your body. However, you've got the recipe there. You've got the DNA code for the production of that breast milk in all of your cells. You ladies have the same DNA recipe for the production of breast milk in every cell of your body, including your skin, your muscles, your brain, your blood cells, your nerve cells. Yet, those cells never produce that breast milk. The only cells in your body that will ever produce the breast milk would be the cells of the mammary glands. And they will only produce it after you give birth to a baby. So, how are these genes turned on and off? How is transcription and translation to produce various proteins from these genes turned on and off? We don't pump out all those thousands of proteins in every cell of your body 24/7, 365 days a year. That would be very wasteful of energy. We only produce the proteins when we need it. So there has to be a mechanism for turning transcription on when we need the protein and turning transcription off when we don't need the protein. And again, much of what we know comes from our study of bacteria. So let's talk about what we do know about the bacterial genome. This fancy word genome just means all the genes in a bacterial cell. Well, they're very simple. Most of what we know comes from our study of Escherichia coli, better known as E. coli. This is the same bacteria we used in our colony transformation lab. It's the same one that lives in the gut of all mammals and is great for us. It's a mutualistic symbiotic relationship as long as it stays there. But much of what we know comes from our study of this little rod-shaped bacillus bacterium. Now, like all bacteria, E. coli has one double-stranded circular chromosome. Only one chromosome, and it's circular, not linear. And all of the genes for the production of the various proteins that E. coli needs is typically encoded in that one circular chromosome. So how big is that chromosome? About 4.6 million nucleotide pairs. So think of that circular chromosome as having 4.6 million steps. Now, do you need to memorize that value? No. Just know that that's much, much smaller than what your chromosomes would have. Within that 4.6 million nucleotide pairs, you'd have about 4,400 genes in that E. coli. A typical eukaryote, we've got a blank here, so think about it. How many nucleotide pairs do you think a typical eukaryote would have in their 46 chromosomes, or however many chromosomes they have? Humans would, of course, have 46. Well, in a human, we have about 3 billion, not million, but 3 billion nucleotide base pairs, so 3 billion steps worth of DNA. So obviously that's many, many more than 4,400 genes. We have thousands of genes on each and every one of our 46 chromosomes. Now, as you can see here in this diagram of E. coli, that one circular chromosome is tightly coiled up to fit inside the cell, but it's not housed in a nucleus. It is housed in what's called a nucleoid region. That's basically just an area where the one chromosome's floating around in the cytoplasm. And remember, when we discussed colony transformation, we said that bacteria have some other smaller fragments of circular DNA that we call plasmids that are also floating around out in the cytoplasm of the cell. Again, these plasmids are much smaller, contain many fewer genes than what that one big chromosome would have. But they are important, especially in transferring antibiotic resistance, because as we learned in our lab, unfortunately, some bacteria do have antibiotic resistance genes very frequently located in some of these plasmids. So when they conjugate and swap plasmids, they pass on that antibiotic resistance to their future offspring. So how do they typically divide? Well, usually we think of bacterial reproduction as being asexual by means of binary fission. So they basically just copy their one chromosome. So that circular chromosome starts to unzip at one origin of replication. Now again, this is a difference from eukaryotic chromosomes. Our linear chromosomes have multiple origins of replication where helicase will start unzipping our DNA in order to replicate it so that it'll just happen a little bit faster. But in these small chromosomes of the bacteria, just one origin of replication, so it's going to be unzipped in both directions around the circle by the helicase enzyme. And then once it's replicated, the cell will basically split in half, and a copy of that circular chromosome will go into each daughter cell. So since you're making identical copies of the chromosome, there isn't any variation in the genetic offspring compared to the parent. The offspring are identical in every way to the parent. And again, remember, as far as survival, at least from an evolutionary standpoint, that would be a bad thing, because variation is always good, and it enhances the odds that some of the organisms will survive if the environment suddenly changes. So this would be kind of the downfall of certain bacteria. The only time you would get variation in bacteria is if by chance there are some sort of random mutations that would happen to that DNA. And certainly that can happen. But that would be the only source of variation in bacteria. In eukaryotic organisms like ourselves, remember the process of meiosis alone. The random alignment of those chromosomes along the equator of the cell and the random assortment produces a lot of variation. That random alignment and random assortment, when the chromosomes pull apart in both meiosis 1 and meiosis 2, creates lots of different genetic combinations in the offspring. And of course we know we can get crossing over in prophase 1 of meiosis 1, which can kind of scramble the genes and produce variation. And we know that the process of fertilization alone creates genetic recombination, because we have thousands of totally different sperm with different genetic combinations, and it's a race to the end to see which of those thousands of sperm gets to the egg first. So meiosis creates variation, random fertilization creates variation, crossing over creates variation, and those simply don't happen in bacteria. Bacteria do not produce eggs and sperm. They do have a method of reproducing sexually, which we're going to talk about in just a moment, but it's not with eggs and sperm. So instead, in bacteria, there are 3 different processes that can create some genetic recombination in addition from the typical mutations that we always see. One is transformation, one is transduction, and one is sexual reproduction, which we call conjugation. So we're going to be taking a look at that. So let's look at transformation first. We talked about Fred Griffith's experiment with the strain R and strain S bacteria, where he learned that when you take dead strain S bacteria and mix it with live strain R, the DNA from that dead bacteria can be taken into the live bacteria to transform it or change it. So transformation is a natural process that happens in bacteria all the time. It's defined as the uptake of naked foreign DNA from the environment. So bacteria can take in DNA from their environment, or in the case of Griffith's experiment, from another type of bacteria in that surrounding environment. So you can see that happening here. This pink strand at the bottom would represent the DNA from the donor bacteria, and here that pink donor DNA is being incorporated into a recipient. And here you can see it's really just a small piece of that donor DNA that's being taken into that recipient. But then when that recipient divides by binary fission, it's going to pass those particular genes from the donor to its offspring. So it now has a new genetic code. That is what happens during transformation, just like with Fred Griffith's experiment. This can sometimes be by way of a plasmid. So this is not showing a plasmid in this picture, but it might be plasmid DNA that gets incorporated into that donor. That's what we did when we did our transformation experiment with creating artificial transformation. We incorporated a foreign plasmid into our E. coli bacteria to transform it or change it. But it does happen in nature too. We have another trans word that can create variation in bacteria, and that's called transduction. When a virus invades a bacterial cell, now that virus would be a bacteriophage virus. Remember when we talked about Hershey and Chase's experiment that proved DNA is the genetic material? Remember that little bacteriophage that had that little hexagonal-shaped head, and that covered up and protected the little DNA core on the inside? A bacteriophage is only going to reproduce inside of bacteria. And when the new viruses are produced inside of the host cell, sometimes bits and pieces of that bacterial DNA will get trapped inside the new bacteriophage's capsid, in addition to the bacteriophage's own DNA. And then when lysis happens, when the host cell ruptures and releases those new viruses that were produced, that little piece of trapped DNA inside can literally get transmitted to new healthy cells and get replicated. So transduction is defined as the transfer of a bacterial piece of DNA into other bacteria by way of a virus. The virus is really just the vector or the carrier that can accidentally transmit the host cell's DNA, in this case bacterial DNA, into another host cell when it continues that lytic cycle. And then lastly, we have conjugation. This is sexual reproduction. This is how bacteria have sex. This is when bacteria literally join their sex pili. They will touch their sex pili together, and they will swap their plasmids. They're small, circular, self-replicating pieces of DNA that just contain a few genes. And whatever genes are in that little plasmid will be transmitted into that bacterial cell through this conjugation sexual reproduction process. Now these terms, F- and F+, the cell that gives away the plasmid is referred to as the F+ plasmid. I'm not sure why they chose the letter F here. It probably has some sort of meaning. But the plasmid that, the bacterial cell that donates its plasmid is the F+ cell, and so that plasmid would be the F+ plasmid. And the cell that is receiving the plasmid is the F- cell, and therefore that incoming plasmid is the F- plasmid. So that's just the way we donate it. So plus is being donated, minus is being received. But when the cell then divides by binary fission, it's going to actually copy those same plasmids and pass it on to the future offspring. So let's talk a little bit about why bacteria do this. Well, we said that often ampicillin resistance genes or penicillin resistance genes or various other antibiotic resistance genes are located sometimes in these plasmids. So when the plasmids, or when the bacteria conjugate and swap their plasmids, they are swapping, unfortunately for us, they're swapping these antibiotic resistance genes. Now again, I say that's bad for us because the outcome is we're seeing more and more bacteria in the environment that are totally resistant to our current antibiotics that we use to treat bacterial diseases. But if you were the little bacterial cell, this would be a positive thing for you because it would enhance your odds of surviving. So unfortunately, by prescribing and using more and more antibiotics, the only bacteria that are surviving and living long enough to reproduce and pass on their genes are the ones that have these antibiotic resistance genes that they continue to swap through conjugation. So we are creating a problem here with antibiotic resistance in bacteria through this very mechanism. Now, sometimes we refer to this process of transferring a gene from one bacterial cell to another by swapping plasmids as transposons or jumping genes. Jumping gene would be kind of the more common term for it, common name for it. Transposons, here's yet again another trans word, is the scientific word for a jumping gene. They can sometimes be called transposable elements. That's down here in the last little bullet. So bacteria, in effect, have jumping genes or transposons by way of plasmids, because these plasmids can be transferred from one bacterial cell to another. Now, later we will talk about how eukaryotic organisms, like ourselves, we have transposons or jumping genes as well. Think about that. We have genes in our chromosomes that for no apparent reason can jump out of one chromosome and into another chromosome. So those jumping genes or transposons can create variation, not only in bacteria, but in us and other eukaryotic organisms as well. So bacteria do have jumping genes by way of plasmids. So how are these genes regulated? We are going to be talking about gene regulation by way of operons in bacteria. And we're going to study two specific operons in E. coli, the tryptophan operon and the lactose operon. So the first operon model that we're going to look at that occurs in bacteria is that of the tryptophan operon. Now let me explain a little bit about what tryptophan is. This is a particular protein that bacteria require to survive, and they need lots of it. They use it for lots of different things. So if there's not enough tryptophan in the environment, the bacteria needing that tryptophan is going to actually try to make tryptophan from other compounds because it needs this particular protein desperately. But if the environment has plenty of it, then the bacteria will not need to make it on its own. So we're going to explain this operon as basically being a type of light switch analogy that explains how the bacterial cell has the ability to turn on transcription and translation and make tryptophan from other compounds when there's none in the environment, and when it can turn off its own production of tryptophan as long as there is plenty in the environment. So we've been throwing around this word operon, and let's talk about exactly what an operon is. I'll come back to this little slide in just a moment. So what is an operon? Well, an operon is a series of genes that codes for production of a certain enzyme. In this case, that protein enzyme is the tryptophan that we've been talking about. And all of these genes are located together, side by side on the DNA strand. And by all of these genes being located in the same part of the DNA, they can be controlled by a single on-off switch. One on-off switch controlling every single gene. It would kind of be like if we had one on-off switch that would control the lights in every room of our home. Same kind of analogy. This on-off switch is actually called the operator. So again, think of the operator as the on-off switch that can turn on transcription and translation of these 5 genes that are necessary to code for tryptophan, or if the bacteria doesn't need the tryptophan, there's plenty in the environment, it can turn it off. So the operator is the on-off switch. Now, so how does this operator work? Well, it controls access of the enzyme RNA polymerase, which is our enzyme that builds messenger RNA during transcription. It controls the access of that particular enzyme to the promoter region. Remember, during initiation, that RNA polymerase that looks like a guitar pick attaches to the promoter region to initiate or start protein synthesis. So this operator, this on-off switch, controls access to that promoter region. If the polymerase cannot access the promoter region, then we know that transcription cannot happen. But as long as it can access it, then we know protein synthesis, transcription, and translation will happen. So the operon is actually all 5 of those structural genes that code for the enzyme tryptophan, plus the single on-off switch that controls it. So let's kind of look at this little diagram, and it may make a little more sense to you. So here is our strand of DNA. Here is our promoter region, and the guitar pick is our enzyme RNA polymerase. You can see here it has attached to the operon. Well, right behind the promoter region, or actually part of the promoter region, is this yellow colored area, which is labeled the operator. This is our on-off switch. And then here are our 5 structural genes that actually code for the enzymes for making tryptophan. You don't need to worry about the letters E, D, C, B, and A. They are just designated by letters of the alphabet, kind of in reverse order here in this particular case. But these are the 5 genes that if they're transcribed into messenger RNA, which is this pink ribbon, and then translation would happen at the ribosome using this messenger strand, then we would actually get the tryptophan. Up in front of the promoter region, in front of every operon, there is another very important gene known as a regulatory gene. And the job of that regulatory gene, labeled here TRPR, R for regulatory gene, is when this particular section of DNA is transcribed into mRNA and then translated into a protein, this protein that it codes for is a special molecule called a repressor. Now, if you think about the word repress, to repress something is to turn it off and shut it down. So a repressor is always a protein that has the ability to turn off or shut down transcription of these 5 genes in the operon. But now look in this particular case, in our tryptophan operon, or TRP operon, TRP is short for tryptophan. Look at the shape of this protein repressor molecule. And I want you to compare its shape to the shape of the operator gene, the yellow operator gene, the on-off switch, in front of all these structural genes that code for tryptophan. Does the shape of the repressor match the shape of the operator? Does it look like the repressor can fit in? Well, you can see that these little prongs on this three-dimensional shape do not exactly match the shape of the operator, so it's not going to plug in there as it is drawn here. So in the tryptophan model, the repressor alone cannot fit. So with the repressor not plugged in to these little slots in the operator, the RNA polymerase has easy access to all of these structural genes. There's nothing blocking its path, so the RNA polymerase can slide down and can transcribe the E gene, then the D gene, then the C gene, the B gene, and the A gene, and all of this messenger RNA would normally be made. So transcription is normally turned on because the repressor is inactive. It's inactive because the shape does not match that of the operator. So tryptophan production is normally turned on because the bacteria needs so much of it. But remember we said if there's already plenty of it in the environment, the bacteria needs to have a way to turn off production of this protein. We don't want to waste energy and time making something that's already in the environment. So let's look at how the tryptophan operon can shut down or turn off or be repressed. Okay, in this particular graphic we have the same operon. Now let's say there's plenty of tryptophan in the environment. The tryptophan molecule here is this little hexagon and pentagon shaped molecule. This is the protein that the bacteria needs. If there's tryptophan in the environment, look what's happening. It will attach to the repressor protein, and in doing so, it will alter the shape of the repressor. See how the shape is changing? The little molecule is bending to a new shape as long as that repressor is attached to it. So now, with the help of the repressor, this protein, the repressor protein, now can plug right into the operator, the on-off switch, which is the yellow portion of our DNA strand. And now that it plugs in, look what happens to our enzyme RNA polymerase. The RNA polymerase cannot get by that blocked operator and can't reach these 5 structural genes that would be down here at the far right, and so transcription cannot happen. So with the help of tryptophan in the environment, the tryptophan attaches to the repressor and alters its shape so that it can fit. It will plug right into that operator and block the passageway of the RNA polymerase and will effectively shut down transcription and turn off transcription because we don't need the tryptophan any longer. So this tryptophan acts as a co-repressor. It cooperates with the repressor, attaching to it and helping it fit into the slots of the operator gene, and therefore shut down transcription. Okay, so I'm going to back up to those slides that I had skipped. Okay. So again, we call this whole tryptophan operon model a repressible model because it's normally on. Normally that RNA polymerase can attach and can transcribe those 5 genes. But if there's enough tryptophan in the environment, it's going to act as a co-repressor and help the repressor attach to the operator, binding to the operator, blocking the way of the RNA polymerase so that it can no longer transcribe the genes. So it's normally on, but it can be repressed or shut down or turned off if need be. If there's plenty of tryptophan in the environment, the tryptophan is a co-repressor, helps the repressor protein attach to the operator and block the passage of the RNA polymerase so that transcription cannot happen. So it's a repressible model as long as it's normally on, but occasionally gets turned off. Okay. So now let's move on to our lactose operon, also sometimes called the lac operon. The LAC is short for lactose. And we've talked about lactose earlier in the year. You know, lactose is a disaccharide sugar. Some people are lactose intolerant and we can't digest it. Well, lactose is a sugar that many bacteria will feed upon, but because it is a disaccharide, it's got to be broken down and digested into its two monosaccharide components, which would be a glucose and a galactose. Those are the two monosaccharides that lactose is actually made of. Well, there is an enzyme called beta-galactosidase that bacteria uses to break down or digest lactose sugar that it can then use for energy. And if there isn't much lactose present, then we would not need this beta-galactosidase enzyme to digest it. But if there is a lot of lactose in the environment, then the bacteria want to use it for energy, so they would need to produce a lot of this digestive enzyme, beta-galactosidase, to actually break it down. So this is called an inducible operon model because the word induce means to start. This operon model is normally going to be off, but will occasionally be turned on whenever the bacteria needs to digest lactose. In normal circumstances, it's not feeding on lactose, but occasionally it needs the energy from the lactose and needs this enzyme to break it down. So when that happens, the operon will need to be turned on or induced. So think repress means to shut down, induce means to turn on. So this is inducible. It is normally off, but transcription can occasionally be turned on when needed. So let's look at the graphic to show this. So we have the same basic setup as we did in the tryptophan operon model. Here is our promoter region where our enzyme RNA polymerase is supposed to attach. As part of the promoter region, we have an on-off switch operator, and then that is going to be followed by in this case, it's not 5 structural genes. I believe it's only 3 structural genes, but these genes code for that beta-galactosidase, the enzyme for digesting lactose sugar. And up in front of this operon, there is going to be a regulatory gene that will code for the production of a repressor. When this gene is transcribed and translated, a protein repressor will get produced. And that protein repressor is going to have some unique 3-dimensional shape. Well, notice in this particular model, this repressor has a shape that plugs in beautifully to that operator. See how the shapes match up? Without anything attached to it. The repressor all by itself magically fits into the operator gene and all by itself can block the passageway of the enzyme RNA polymerase. So since it cannot get by this blockage, the enzyme cannot get down to these structural genes to actually produce the enzyme beta-galactosidase. But if the bacteria needs to digest the lactose sugar to produce more energy, there needs to be a way to turn this thing on. And the lactose sugar itself in the environment will serve as what we call an inducer. Now, if you look at this graphic, we have the same operon as we saw in the previous slide. But the little green circle is what's new and what's been added. This molecule, represented by the green sphere, is lactose, or this is calling it allolactose. That's just a variety of lactose sugar. So if there's lactose in the environment that needs to be broken down and digested, the lactose can attach to the repressor. Now watch what happens. When it attaches, what happens to the shape of the repressor? Boom, there's a shape change. The repressor has changed its shape, and now is that repressor going to be able to plug into the operator? No, you can see the little prongs are bent outward. It doesn't match any longer. So now the repressor cannot attach to the operator, And so nothing is plugged in here to block the access of to these genes. And so now our RNA polymerase can slide down and can transcribe these 1, 2, 3 structural genes that make beta-galactosidase and these other enzymes for digesting lactose sugar. It's only going to make these enzymes when there is lactose present. So the lactose behaves as an inducer. An inducer is a molecule that will start transcription. To induce something means to start it. It will start transcription because by attaching to the repressor, it alters the shape of the repressor so that it is inactive and can't fit the operator anymore. So these two operon models are basically opposite. Okay, we will be modeling these in class, so hope this is helpful. You may want to go back and look at this again and study these models so that you can understand the difference between the repressible model that's normally turned on, can be repressed or shut off, and the inducible model that is normally on and can be shut off. I said that backwards. Inducible model is normally off and can be turned on.